So this is our chapter molecular basis of inheritance. Today we'll study the cell and features of human genome. As you know, in previous chapter we have done the goals and aims of human genome project and methodologies of human genome project. Today we'll study about cell and features of human genome. So these are the salient features of human genome. First one is human genome contains 3164.7 million nucleotide bases or base pairs. Next one is the average gene consists of 3000 bases but sizes vary greatly. Total number of genes is about 30,000. 21,000 are protein coding and 9,000 are non-coding RNA genes. Some 21,000 pseudogenes are also present. Previously, it was believed that human beings carry 80,000 to 1,40,000 genes. The size of genome or number of genes is connected with the complexity of body organization. Lily has 18 times more DNA than human genome. In spite of this fact, Lily produces fewer proteins than a human being. It means that it's having more introns than exons. The function of over 50% discovered genes is unknown. अभी तक हमें नहीं पता कि उनका क्या काम है. Less than 2% of the genome contains coded information for protein synthesis. So by differential splicing, a gene may code for 2 or more proteins. As I told you in the previous lecture that chromosome 1 has maximum number of genes, that is 2968, while chromosome Y has minimum number of genes, that is 231. They are the maximum and minimum genes for the human chromosomes. 99.9% of nucleotide bases are exactly similar in all human beings. The length of different human genes vary widely. The average gene size is 3000 base pair. Beta globin or insulin gene are less than 10 kilo base pairs long. The largest gene of human body is that of Duchenne muscular dystrophy on X chromosome. It is 2.4 million, 2400 kilo base pairs long. The smallest gene is transfer RNA gene with 76 nucleotides. So part of DNA which contains repeated sequences is called satellite DNA. Satellite DNA is of several types. It's one of them is moderately repetitive sequences. There are mini satellites and there are microsatellites. Microsatellites are basically they have 5 to 8 pairs, base pairs, and mini satellites are having 11 to 60 base pairs. So these sequences are clustered around centrosome and near the ends of chromosomes. These repeated sequences are called junced DNA. Repeated sequences have no direct coding function. They however provide information about chromosome structure, dynamics, and evolution. So about 1.4 million single base DNA differences or single nucleotide polymorphism, which are commonly called SNPs, have been identified in humans. Their number may be more than 10 million. This will be helpful in finding chromosomal locations with disease associated sequences and tracing human history. These are the applications and future challenges of human genome. First one is disorders. More than 1200 genes are responsible for common human cardiovascular diseases, endocrine diseases like diarrhea, neurological disorders like Alzheimer's disease, and cancers, and many more. And then, just like cancers, efforts are in progress to determine genes that will change cancerous cells to normal. Uh, it will indicate prospects for a healthier living, designer drugs, and then genetically modified diets, and finally our genetic industry. Then it will be possible to study how various genes and proteins work together in an interconnected network. All the genes or transcript in a particular tissue, organ, or tumor can be analyzed to know the causes of effect produced in it. And then information about natural capabilities of non-human organisms can be used in meeting challenges in healthcare, agriculture, energy production, environmental remedies. For this, a number of model organisms have been sequenced. For example, bacteria, yeast, Drosophila, fruit fly, rice, etc.